students, I hope all of you are doing very well, and after learning the basics of immunity, how our immune system is working, from today I want to tell you in depth about the working of our immune system. In a previous lecture, I hope in some of the slides I have mentioned slightly about the immunoglobulin. So, what is immunoglobulin, how they are working, everything I will tell. In a previous lecture we have seen about the adaptive immunity, B cells and T cells, and humoral and cell mediated immunity have seen, and we also have seen the differences and how they are coordinate. So, today I will tell you about the immunoglobulin. Antibodies and immunoglobulin both are many of the times we actually use those similar these words are in a very similar manner. So, I will tell you detail about what is immunoglobulin, the introduction. I will tell you about the structure and how they are actually differentiated, various types of immunoglobulin, and I will tell you how this antibody is actually working, functions including monoclonal antibodies, and similarities between antibodies and immunoglobulins. See, the word antibody and immunoglobulin, both the words actually we are actually using interchangeably, okay, and they are similar and there are some differences. I will tell you more in detail. So, in my previous lecture I have mentioned about the humoral immunity and cell mediated immunity. I have told you that humoral immunity, it is actually generated by the B cells, and I also have mentioned that B cells are working based on the antibody. So, here the entire concepts are actually getting clear. So, the humoral immunity is actually mediated by the special group of antigens binding protein are called immunoglobulin or antibodies produced by the B lymphocytes. Many of the books you will be getting that antibody and immunoglobulin both are same, but some books the modern scientists they are try to differentiate between both the things, but for our purposes we shall use the word interchangeably. This is Y shaped protein, it can detect, bind to the harmful specific substances like antigens like bacteria or foreign substances. Here you can see, can you see here is some Y, it is look like Y, na? Y, that is why we call it as a Y shaped. So, this is the modern understanding of the immunoglobulins. So, it says that all the antibodies are immunoglobulins, but all immunoglobulins are not antibodies. We are actually use both the word synonymously. Okay, you should keep this word in your mind, but for our understanding, to understand how our immune system is working, we should use this word synonymously. Okay. So, since the majority of the antibody types are found in the globulin fraction, that is why they are actually called immunoglobulin. I hope now you are slowly able to understanding. Majority of these antibodies are actually found on the globulin function. That is why those antibodies are actually called as also immunoglobulin. In 1931, Thyselius and Kevet, the scientists, they have isolated antibodies from the blood plasma by electrophoric mobile assay. Okay, and after that WHO in 1964 they have coined the word called immunoglobulin. The production of antibodies are also carried out by the B lymphocytes. You are already aware about that thing, because in my previous lecture I have mentioned that what are the activity of B cells, how B cells are producing antibody. Because of the presence of antibody, our viruses are not able to work in our body and they actually get destroyed. So, immunoglobulins can either be membrane bounded B cell receptor or secreted like antibody. So, here is a very interesting thing. So, what we have seen in our previous slides, here you have seen that Y shape. So, here in the structure I have, you can see this is the Y shaped, I have tried to give you more detailed the structure. If you see the structure of the immunoglobulin, you will to identify that it consists of the two identical heavy chains and two identical light chains, H for heavy chains, L for light chains. See, heavy chains, this green part is actually heavy chains, okay, and this part is actually red part actually light chain. All the kinds of antibodies you will be getting this kinds of thing. Okay. So, what is the molecular weight? Its molecular weight be 55 that is 1000 Dalton to 75,000 Dalton, and in this case its molecular weight will be 3000 Dalton. So, that is why it is actually called light chain. Its size is much more smaller and its weight is much more smaller. Okay. And antigen binding site is this one. This is the antigen binding sites. Because of their variation for various kinds of bacteria, for various kinds of viruses, we are getting huge different kinds of antibodies. Okay. Each heavy chains contain approximately 450 amino acids, and light chain obviously it is a small and its weight is much more smaller than a heavy chain. That is why only 212 amino acid almost amino acids will present in this scenario, and there are 5 different types of immunoglobulin, okay, denoted by the alpha, delta, epsilon, gamma, and that is mu. I will tell you more what are the various types, and these types of okay, I have already mentioned these things, and let me tell you another thing about their variable region. Okay, both the types of the chains are actually composed of the 2 regions. One is the variable region. See this part it is the variable region, they can change. There might be some variation can be possible, whatever the things you are observing now, there some variation is possible in those areas. Why this variation is possible? Because in my previous lecture I have mentioned you that there are huge amounts of viruses are present in this our universe. There are lots of viruses are to be discovered, they can enter, they can destroy our cells. So antibodies are actually microbe specific, antibodies are actually microbes specific. That is why some variation actually happening in this scenario, because this is the area where it will be go to the bind with the, or it will be work, it will go and bind with the microbes. So that is why this binding site here some variation is actually possible, and in light chain in other area this will be the constant regions. Now these regions that change various structures depending on the differences of antigens are called variable regions, whatever I have mentioned, and the regions that has constant structure is called the constant region. There are 5 different classes of immunoglobulin. One is called IgM, one is IgG, one is IgA, one is IgE, and one is IgD. See, this is the bigger, this is the much more bigger than other kinds of types of the immunoglobulin, the IgM. Okay. So, from where the M is always coming? M is actually coming from here because of the mu chain. Okay. So, G is actually coming from here because of the gamma chain, and IgA is here, and from epsilon the E, from del it is D. Okay. So, from Greek alphabet these similar names are we actually getting. So, you should remember this picture, because it is very important for you, because in examination there might be chances that your teacher might be asking a question that what are the various structures of IgG, IgM, IgA, IgE, and IgD. You should remember this structure. Okay. So, what is the simplest way to remember the structure? M means it will be bigger. So, if you try to remember M, it will be much more bigger. IgG it will be Y, IgE it will be also Y, IgD it will be also similar to Y, but for A it will be Y opposite Y opposite, you should remember in this manner. But in both the cases, in D, E and G, it will be slightly similar, but yes, there are some differences. I will tell you more what are the differences here you can see. So, if I tell you more about their working, their percentage in our body. So, what is the function of IgM? Main antibody or primary responses based in the fixing complement. The monomer form the IgM serve as the B cell receptor, and how much percentage it is present in our body? 6%, very less, not so much high. IgG monomer, the main blood and antibody, secondary responses neutralize the toxin and opsonization. It is the highest, 80%. So, maximum cases you will be getting IgG. Many of the testing kits they are actually using presence of IgG, because it is the highest amount of immunoglobulin present in your body. And IgA means Y opposite. If you just rotate this, it will look like this Y. So, what is the function of this IgA? It secretes into mucus, tears, saliva and colostrum, and 13% of in our body we have this kinds of immunoglobulin. IgE monomer, antibody of allergy and antiparasitic activity, it is providing IgE. Very less amount of immunoglobulins are actually IgE. and IgD monomer, they are B cell receptor only 1%. I hope it is very much clear to you all. Let me tell you more. Heavy chain, already you are aware what are the kinds of heavy chain are actually present. They are nothing but they are Greek alphabet for mu, m, gamma, g, and alpha A, aspirin E, and del D. Number of binding sites, it has highest 10, and it is 2, 4, 2, and this 2. Molecular weight. In case of molecular weight, obviously you have seen the picture of the various kinds of, let me take to you again, yeah, you have seen the picture. This is the bigger, hai na, out of this all the things, all the 5 kinds of immunoglobulins. This is bigger, so that is why its molecular weight obviously it will be much more bigger than the other one. And IgG that is 1,50,000. This is also much more bigger, IgA, because it is Y opposite, hai na, double. So 3,85,000 Dalton, and this one is 2 lakh. This is 1,80,000. Cross placenta, no, they cannot, and it is yes, and both the things are cannot pass in the placenta. And fixus complement, yes, it is possible for IgM, and IgG, yes, it is also possible, but for remaining things it is not possible. Fc bands, here is no Fc bands. Phagocytosis may happen here, and it is also possible in the mast cell basophils. Now, the biggest question. Sir, you already have covered, you already have discussed about the immune system. You already have mentioned that immune systems are having, we have three types of 3 layer of defense. You also have mentioned that immunoglobulins are and immunoglobulins and antibodies were actually using both the word synonymously. Now, sir, how antibodies are working? You might be thinking, right? So, here is a very beautiful picture how antibodies are working. So, for example, this picture, let me again draw here. This picture, it is so similar with SARS-CoV-2 coronavirus, hai na? What is it will happen? So, at first the virus will bind to the receptor cell surfaces. We have various kinds of antigen presenting cell, I hope you remember, hai na? So, it will virus will bind to the receptor cell surfaces like this, you can see. Then receptor mediated endocytosis will be happening. Endocytosis means it will be engulfed, it will take. Then acidification of endosomes and endocytosis trigger the fusion of the virus into the cell and entry of the viral DNA. Viral DNA entered in that cell, and then antibody block the binding virus receptor and can also block the fusion event. So, when these things are happening, similarly another work is also happening. This is the called antibody production. When in your cell surfaces these kinds of activities are actually happening similar at the same time, before happening this work actually coming from here. When the virus is entered, it is actually endocytosis and other activities are happening. At the same time, because of its enter, some that is a communication was happened within your immune cells, and your B cells are actually producing, they are signaling, helper T cells actually signaling to the B cells, and they are releasing some lots of antibodies. So, first binding of the antigen was happened, then your B cell was actually activated because of the chemical signal done by the helper T cells, and because of that signal B cells will become the plasma cell. B cells will become the plasma cell, and plasma cell will generate lots of antibodies, and that antibody will go, and for future viruses that antibody will surround. ये जो देख रहे हो ना virus, this virus you are observing here, this virus actually gets surrounded by the antibodies. Now, for other kinds of your engulfing cells, dendritic cells, other cell, it will be very easy to engulf the entire complex. This is how our antibodies are actually working. Let me again remove all these inks from my slide so that you can understand. So, this concept is very simple. Viruses entered in your body, your cell surface and your other immune cells actually got exposed, and they have presented that exposure called as an antigen presenting cell. And after that, your helper T cells actually activated your B cells. B cells produced huge amount of antibodies. Some viruses actually destroying your cells, and the remaining viruses what are actually present, they will not able to destroy because of the antibodies. Now, we might be wondering, सर अगर ये प्रोसेस इतना सिंपल है तो कोरोना वायरस के केस में व्हाई वी फेल टू डिफीट कोरोना एट द इनिशियल डेज। सन 2021 में जनवरी 2020 की जनवरी में जब ये फैला था कोरोना, एट दैट टाइम व्हाई वी हैव फेल्ड, व्हाट यू आर सेइंग, इफ इट इज सो सिंपल देन व्हाई हैव फेल्ड। I hope you remember in my last lecture I have mentioned that the antibody production, it is not a simple work. For that you need some time, minimum 7 or highest 14, but it depends how our immune system is strong. If some person's immune system is very strong, in that case there will be very lethal immune response. SARS-CoV-2 will not able to invade his immunity. You have seen lots of people, there are lots of stories when we have seen that people are actually not infected by coronavirus despite they got exposed very rapidly, okay, but unko corona hua nahi, they are working in hospitals. Why? Because of the strong immunity. That is why people are actually suggesting that your immunity should be much more strong. Now, how antibodies are working, another simple picture. In my previous slide, here I have mentioned the production and the mechanism. Here I am to tell you, in this slide I am to tell you. So, in future again if the same virus may enter in your body, your immune system will be work, it will be destroyed. Kaise? For example, measles virus, measles, it is an antigen. If measles virus enter in your body and you previously got exposed by measles, at that scenario in your body you have existing antibodies, you have existing antibodies. So, what will happen? You can see this Y shape things will very nicely fix here, very nicely fix here, and because of the fixation it will be nicely fixed here. Because of this fixation your other immune cells will be activated and it will engulf and destroy. Okay. You will be never get affected by the measles. Similarly, for flu viruses also you can see this Y shape antibodies can very nicely bind here. When it will bind here, other cells, our other that is human cells will be got to know ki yes, there is some pathogen is present, our B cells got trapped them, we have to engulf them, we have to kill them, we have to remove them. Now, similar case chickenpox also you can see. I hope another thing you are observing that, let me remove the ink. I hope another thing you are observing that all the antibodies are actually Y shaped, है ना? देखो, this is also Y shape, this is also Y shape, this is also Y shape. Where is the differences? Differences is the here. Differences is present in the here. That is the binding site. Okay. Because of this such differences, huge number of antibodies we can create in our body. Okay. So, if there is new pathogen, if we do not have antibody, it will be dangerous for us, our body will have to generate the antibody. So, for SARS-CoV-2 similar manner what I have mentioned you. So, antibody will be blocked to the spike protein, and then when spike protein will be get blocked and your macrophage will engulf and it will destroy. So, what are the functions of antibody? So, IgG provides the long term protection because it persists for months and years after the presence of the antigens that has triggered their production, and IgG protect against the bacteria, virus and neutralize those bacterial toxins, and including the main function of IgG is to the bind antigen on the macros before they invade the tissues. Okay. And if I tell you more, IgA are the first defense for the mucosal surfaces such as intestine, nose and your lung. Okay. Because intestine, nose and lung, these are very easily get exposed to the nature. Coronavirus जैसे हमारे body में enter करेगा ना, when coronavirus will enter in our body, first these cells will be these organs will be come in the contact. So, that is why IgA will be the first defense, and then IgM, it will also involved in the ABO blood grouping system and surface on the RBC, and IgM enhances the ingestion of the cells by phagocytosis. IgE can bind to the mast cells and basophils with which precipitate to the immune system. Some scientists also think that IgE's purpose is to stop parasites, they can stop parasites activity, and IgD it present on the surface of B cells and plays a role in the induction of the antibody production. Now, after discussing a lot of things, another very concept called monoclonal antibody. You might have heard this word lots of time, monoclonal antibody. Due to the shortage of time, I will not discuss monoclonal antibody today, because I have a different I have a separate lecture on hybridoma technology. Okay, when I will discuss with the hybridoma technology, I will tell in detail about the monoclonal antibody. For today's understanding, you have to just understand that they are actually antibodies, mono means single, only one antibody. We are actually using these monoclonal antibodies in various kinds of diseases like cancer. Every year you can see almost every 4 or 5 years there are lots of new drugs. There are lots of new drugs are actually coming to the market. MAB, MAB at the end, where you will see MAB is written, you have to understand that it is monoclonal antibody. So, lots of monoclonal antibodies are actually come to the market, which are actually working as an antibody. Here I have listed other drugs also. So, another thing actually called polyclonal, monoclonal and monoclonal antibody cocktail. I hope many of you have heard that Donald Trump, US president, when he was infected by COVID-19, he the doctor given him one cocktail, antibody cocktail. Few months ago in our India also one cocktail was actually launched. Its price was I think 58,000 for one dose. Okay. So, for monoclonal antibody only one antibody will be there, and in this case multiple antibodies will be there, and here it will be cocktail, it will mix, it will be cocktail. I will tell you more, do not worry, I will tell you more in my upcoming lectures also how they are working. Now, what are the similarities between the antibody and immunoglobulin? So, immunoglobulins and antibodies are the disease fighting proteins developed by the most vertebrate in response to the particular antigen. Already have seen both the immunoglobulin antibodies are component of the immune system. Both immunoglobulin antibodies are actually glycoprotein, and both are actually similar region in the in their molecules. Immunoglobulins actually attached to the B cell membrane, while antibodies actually floated around in the circulation. That is the main difference. You can also tell immunoglobulins actually attached to the B cell membrane, and they are actually floating around the surface. So, both immunoglobulins and antibodies are comprised with the heavy chain and light chain, and both immunoglobulins are and antibody comprised the variable and constant region. I hope you can remember we have seen in our Y shape protein this region is the variable region. Both of these are actually produced in response of the presence of the antigen, and both of the things actually can be found in the secretion as well as in the circulation, and both the immunoglobulin antibodies involved in the protection protecting against various kinds of pathogens. Okay. These are the various similarities, but what are the differences? Differences already we have learned, but still let me just tell you. So, immunoglobulin refers to the class of structurally repeated protein in the serum and the cells of the immune system with specific antibodies. Antibodies refers to the globin protein, which is the produced by the B cells responding to the particular antigen. So, their activity is same, but they are in terms of their structure, their origin, there are some differences occurs in the B cell, and you can freely found in the circulation. Okay. And has a transmembrane domain in order to attach to the plasma membrane B cells, and it does not have any transmembrane domain. 5 class, what are these things? IgG, IgM, IgD, IgA, IgE, and a particular antibody is specific for a particular pathogen, and functions depend on the type of the heavy chain, and they are non-self antigen recognized by the specific antigen, and they are neutralized by the antibody. Let me tell you more. Immunoglobulin has a transmembrane domain. We already have seen in that in our previous slide, and they do not have the transmembrane domain for antibodies. And if I tell you more, apart from the antibody globins, the immunoglobulin class also contain other globins such as cryoglobins, microglobins, and abnormal myeloma proteins. Majority of this antibody are actually found in the globin function, and antibodies are actually called as a immunoglobulin. Okay. And officially WHO coined this term in 1962, and in 1931 the first time the antibody was actually identified. So, all the antibodies are actually immunoglobulins, but all the immunoglobulins are not antibodies. You should remember these words. Okay. So, we are actually use both this word synonymously. Many of the books they have not distinguished between both these words, both this immunoglobulin and antibody. I wish that you are learning B Pharm, you are a undergraduate student. So, you should learn in a very most updated manner. That is why I have added the differences, though it is not a part of your syllabus. So, today we have learned about the introduction to immunoglobulin, structures, types, how antibodies are actually working, their function, monoclonal antibodies, similarities and their differences. So, in my next lecture, I will tell you more about the other kinds of our immune cells, how they are working. So, stay with us, and I am deeply thankful that AKtU is actually organizing such digital education for you, so that you can learn in a very advanced manner. So, thank you all of you for being with me, and I hope I was able to give you a very good insights. So, in our next lecture we shall learn more. So, thank you all of you. God bless. Jai Hind.